Hello to all the HubLE audience. My name is Carmen Huesa. I'm a postdoctoral research fellow at the University of Edinburgh. I am here today for an edition of the Meet the First Author with Nick van Gastel. Did I say it right?
Yes, you did.
We will be talking about his paper titled Lipid Availability Determines Fate of Skeletal Progenitor Cells via SOX9, which was recently published in Nature. So, Nick, to start with, could you tell us what a summary of your paper and how you think it fits in the big picture?
Of course. So the question we started off with for this study was how differentiation of skeletal progenitor cells during bone healing is regulated. And so what we know, and it might be good to explain this for the audience, what we know is that there's two different ways bone healing can progress after a fracture. If there is a very small fracture or if it is a very stable environment, you will get direct bone formation. So the progenitor cells will differentiate directly into bone-forming osteoblasts. In contrast, if it's a larger fracture or if there's a lot of movement at the fracture site, you get a two-step process where first the progenitor cells form cartilage, which sort of serves to stabilize the fracture site and which is then later replaced by bone. And so we have been, or the field has been studying the differentiation of progenitor cells into osteoblasts and chondrocytes for quite a while now, and many of the cytokines that are known can both influence osteogenesis and chondrogenesis. For example, the BMP family or different TGFs or fibroblast growth factors can both influence osteogenesis and chondrogenesis. And so what we wondered was, are there any other signals coming locally that will influence whether a progenitor cell will become an osteoblast or a chondrocyte? And so one of the things that we know when we started the study is that an interesting feature of cartilage is that it's avascular. There's no blood vessels in cartilage. And so what we wondered is, could that play a role? Is that just a consequence of cartilage, or could that be a cause as well, that when blood vessel availability is limited, that cells are more prone to become chondrocytes? And so to test this, we used a bone fracture model in mice where we could actively block blood vessel ingrowth into the periosteal layer. And so the periosteum is the thin layer of cells on the outside of the bone that contains the progenitor cells that will repair the fracture. So what we found basically is that if you block blood vessel ingrowth, you get cartilage formation in sites where you normally get bone formation. And so really showing that absence of blood vessels can act as a cause for chondrogenesis and not just a consequence. We then took it in vitro to find out more what the signals could be coming from the blood vessels that are triggering this switch. And because of the interest in the lab in nutrient signaling, we looked at different nutrients and how they might influence differentiation of skeletal progenitor cells. So we isolated periosteal progenitor cells and then exposed them to low oxygen, low glucose, low glutamine, and low serum. And surprisingly, we found that the biggest trigger was not low oxygen as we expected, but low serum. We could then further narrow this down to the absence of lipids and found basically that if you have low lipid availability, a skeletal progenitor cell will turn into a chondrocyte. And more mechanistically, we could link this to FOXO transcription factors. So these are well-known stress response factors, and we find that they are also involved in the immediate response to lipid deprivation stress and directly bind the SOX9 promoter and activate it. So I think it sort of adds a new aspect to the differentiation process of progenitor cells to either osteoblasts or chondrocytes, where we're no longer just have to think about different cytokines and growth factors that influence this process, but also about the role of nutrients that might be very different in the local microenvironment, and that can also influence this process.
Yeah, that is a brilliant summary of the paper. In the discussion at the end, you say of its possible applicability in the field of osteoarthritis. Could you maybe expand on that?
It's more of an interesting observation that I think is definitely an avenue for future research. So if we look at the four of the key events in our process during fracture healing, is that you have changes in vascularization that are linked to changes in lipid availability that are then translated into activation of FOXO and downstream SOX9. And so if you look at osteoarthritis, these four different aspects have all been involved in aspects of osteoarthritis. So you have increased vascularization, for example. There is dysregulation of FOXOs. There have been some reports that say that there is an increase of fatty acid oxidation, and SOX9 levels are very well known to be dysregulated in this process. And so what we think is maybe this pathway might also be dysregulated in osteoarthritis, and maybe targeting lipid metabolism might be a way to have a new approach that maybe you could think of it maybe as sort of a vicious cycle. And I think that's what people, I'm not that familiar with the osteoarthritis field, but I think it sort of seems to me like a vicious circle where you have vascularization that in the end leads to cartilage breakdown, which then triggers more vascularization. And maybe by interfering with lipid signaling, so that even if you have vascularization, the chondrocytes are not seeing these lipids and are not losing their identity, that could give some new opportunities. So I think that's definitely something that for future research will be worth looking at.
I think so too. I think it's really, really worthwhile. It's given me a few ideas, actually.
That's what we hope to get out from the paper, that people will start looking more at nutrients as well in other parts of the skeletal field. So yeah.
I mean, the more we look at diseases like of the musculoskeletal tissues like osteoarthritis, the more complex it becomes. Mm-hmm. And it is very clear that these cells, both the osteoblasts and the chondrocytes, are highly complex cells that use a lot of different pathways and have a very complex biochemistry. And we need to look at the full picture to be able to try and sort out the problems that we encounter in disease. Mm-hmm. Yeah. So just to continue the discussion, I actually quite like asking people about how the experimental approach was. What was difficult? What was not difficult? What was, a sail through? If you could just give us like one complicated experiment that you thought was difficult, and then we'll move on.
Okay. I think for difficulty, what we struggled with a lot was to translate our in vitro findings to an in vivo setting. And I think that proved to be really tricky because we weren't able to really locally manipulate lipid levels in vivo. And so our biggest finding is that local availability of lipids can regulate cell fate decision in progenitors, and we couldn't manipulate really local lipid levels. And so because we couldn't decrease the lipid levels to see if that would lead to more cartilage, we did the other way around, and we tried to increase lipid levels locally to see if that would then have the other effect of inhibiting chondrogenesis. And that is what we found. And so that, I think, was really tricky to sort of find a way to really show that this is something that happens in vivo. And I think the only way we could do really the deprivation was to use metatarsal cultures. And I think I still love that model. I think it is an amazing model to look at these early events, and it might not really be fracture healing, but a lot of the same processes as you have during fracture healing are also happening during development. And I think the metatarsals are a beautiful model. And so I still found that very striking, that the moment you take the serum out of those cultures, or you put them on low serum, there's no osteoblasts that form at all. There's zero. In any of the slides we looked at, we couldn't find any. And you just add back fatty acids and they all come back. So I think that was, although it is not really an in vivo, it's sort of a semi in vitro ex vivo experiment. I still think that was one of the most beautiful experiments we did. And I really have to thank some of the technicians from the lab in Leuven that really have these fine hands to dissect out the metatarsals and make these beautiful cultures. So that, I think that was one of the it's tricky, but one of the most beautiful experiments in the end.
I've done the metatarsal protocol myself. It's beautiful. You definitely get really robust data from it, and very reproducible, which is brilliant. Yeah, it was a brilliant experiment. Overall, the paper is amazing in my opinion.
Thank you.
So just a bit more. What do the results mean to you? You think?
You mean more for me as career-wise, or for like the field direct research?
Direct research. What is it that you think this means for you to take forward, and what the outcome of this is going to be for you?
So I think one of the things that I'm really interested in is how metabolites are really performing signaling functions. And I think that is something that also now with my work that I'm doing now as a postdoc at Harvard is more focused on that in the blood system, in blood stem cells, but really trying to get a grasp of how our metabolites, such as lipids, for example, really playing a role as signaling molecules, how are cells sensing them, and how is this influencing cell fate decisions. And so I think it sort of adds a whole new layer of cellular signaling and communication that we sort of weren't really aware. I mean, we know metabolism is important, but the role this plays in these essential like decision-making events was not really known. So I think that is something where I personally really want to dive further into and try to understand how cells are really sensing these kind of nutrients, and how in different systems you might have different ways that these very simple molecules, because a metabolite is biochemically much more simple than something as complex as a growth factor or a cytokine, for example, and how these very simple ways for cells of talking to one another can play a role in important cellular functions. So I think that's sort of what I find really exciting about this, and what I hope to further look into in different systems, at how we can manipulate this, and how we can use that for regenerative medicine, or to prevent tissue degeneration, or even in the setting of malignancies such as cancer, for example, where these processes are known to be dysregulated.
Yeah, that's one of the diseases I was thinking of as well.